Hello, I am Dr. Stacey Dutton. I am a neuroscientist, and I'm going to talk to you today about the COVID-19 vaccine. And so I'm going to specifically focus on the science behind how these vaccines work, and hopefully that will help clear up any misinformation that there may be in regards to the mechanisms behind these vaccines. And so I will be remiss if I do not mention part of the reason why many persons of African descent are apprehensive about engaging with these vaccines. And a large contributor to that is just the history of abuse that the medical and scientific community has had on these communities, in addition to just our lived experiences. And so of significance would be the Tuskegee experiment, where Black men who had syphilis were denied care so that the medical professionals and scientists could better understand the trajectory of the disease. Also, we could talk about Henrietta Lacks, a Black woman who had cervical cancer, and her cells were taken from her without her consent and then used as the main cell line for many scientific experiments that led to many advancements. And then we can also talk about Dr. Marion Sims, who is the father of modern gynecology, and the fact that he did most of his experiments and most of his studies to develop techniques on enslaved Black women. And also just the general experiences that Black people tend to have when they engage with the medical community. So all of that really forms the foundation as to, despite us being disproportionately impacted by COVID-19, there still is a distrust in the medical industry and wanting to engage in getting this vaccine. And so the purpose of this video is not necessarily to convince you to get it. It's just to provide you the foundational understanding as to how it works and hopefully allow you to make the best decision for you. And so with that, it's important that I first talk about some important terms because they will be essential for understanding how this vaccine works. And so the first is virus, right? Oftentimes in just general jargon, we hear the term virus thrown around, but I just want to make sure that everyone is clear on what a virus actually is. And first, I want to mention that a virus is just an infective agent that typically consists of a nucleic acid molecule in a protein coat, is too small to be seen by a light microscope, and is able to multiply only within the living organisms of a host. Now that seems very scientifically jargony, right? And so let me just break that down for you in a more layman's vocabulary here. So viruses essentially are not, we don't necessarily consider them to be living beings in the sense of them being able to do specific things that living organisms do. So as scientists, we have 8 characteristics that generally classify a organism as being living. A virus does not necessarily meet all those characteristics. And so it's actually considered a moiety. And specifically, all a virus is a type of molecule known as nucleic acid. That's really all it is. It's wrapped in a protein, and that in order for the virus to live, it needs a host cell. So it needs either the cells of humans or another organism to invade and there allow its nucleic acid to multiply. And so that's how it's able to, in a sense, live. And so when it invades our cells, it essentially hijacks the machinery that our cells use to divide. And so there are different types of viruses, okay? And the particular strain of, or the particular species, I'll say that just for general terms, that we're dealing with right now is a coronavirus. And a coronavirus is actually one of the most well-studied class or species of viruses. And it's been studied since the mid-1960s. And so there are about 7 different strains of coronaviruses that can affect humans. Most of them are pretty harmless. 4 of them actually just encode the common cold. And so at some level, all of us have interacted with a coronavirus before. But 3 of them are actually quite more critical and can lead to more serious and dangerous conditions. So the first was identified in 2003, and that was the virus is SARS-CoV or SARS coronavirus. And this causes what is known as severe acute respiratory syndrome, which is SARS, okay? And then in 2012, MERS was identified on MERS-CoV, which is the virus is MERS-CoV. And it causes the Middle East respiratory syndrome. And this was also another severely impacted coronavirus. And so then lastly, that brings us to 2019 when SARS-CoV-2 was identified, which is basically the coronavirus disease. And so I also wanted to just note that this jargon can be a little bit confusing, but I just want to note that the SARS-CoV-2 is the virus and COVID-19 is the disease. So when you get tested for whether or not you have the virus, the test that they're running actually is looking for the presence of the SARS-CoV-2 virus, okay? It's not necessarily looking for COVID-19 because COVID-19 is the disease. And so if you have the virus and you begin to show symptoms of the disease, then you're termed as being COVID-19 positive, okay? So if you have the virus but are not presenting symptoms, you technically don't have COVID-19. You're considered to be asymptomatic. But the concern with that is that you can still spread the virus because you still are impacted with it, though you may not be exhibiting symptoms. So that's in part why this is such a critical situation to try to get a hold of, because if you are not aware of your status and whether or not you have been exposed to the virus, you still run the risk of exposing others and, further amplifying the severity of this condition, with the pandemic more so. So here is a cool schematic that basically provides you an understanding of how the SARS virus works, okay? So this image here is essentially the COVID-19 virus, or the SARS-CoV-2 virus. And I want to point out some unique features about this virus, okay? And so on the external surface of it, you see these spikes. These spikes are extremely important as it allows the virus to be able to penetrate and get into your cells, okay? So inside the cells, though, you see this red, squiggly line. So that red squiggly line is representing the genes or the genome of that particular virus. It's important to note that the genome of the COVID virus or of the coronaviruses is RNA. So use RNA as a genetic material. So as I mentioned, these spikes are extremely important as they bind on a receptor that is found on our cells. This receptor is the AC2 receptor. And once the spike protein binds to that receptor, it allows the virus to now enter into the cell. Once it's in the cell, as I stated before, the virus functions by using our body's own replication machinery to produce more of its, excuse me, to produce more of its genomic material. And it continues to do that until the cell erupts, dispersing the viral material now within your body, providing it the opportunity to now go and infect more and more cells. And so in short, this is how the virus works. So then let's talk about the vaccines. What are the vaccines and how do they work? So again, I want to talk about some important concepts before I, bring the story all together. And the first, I want to make sure that people are clear on what vaccines actually are, okay? So vaccines are essentially treatments that are used to, in a sense, exploit our body's immune system. It exploits how our immune system naturally functions. So when you are exposed to some sort of pathogen, okay, and it invades our cells, our body is able to generate antibodies. And this is a pretty complex process, and I'm not going to go through that in this particular discussion, but our body is able to generate antibodies so that if we are ever exposed to that pathogen again, our body has already developed the defense so that you can adequately fight off the particular pathogen. And so vaccines function in the same way. Essentially, what they do is they, in a sense, provide you with either weakened versions of the virus or small fragments of the virus. So you're injected with that, and that triggers your immune system to mount a response, and therefore ultimately resulting in the development of antibodies so that you have this natural protection against being able to fight off this pathogen if you're ever exposed to it again. And in general, vaccines are extremely effective. It is because of vaccines that we have been able to achieve herd immunity for a lot of infectious diseases in this country. And so then the major point of discussion is the new vaccines that have been developed in order to treat COVID-19, okay, or in order to protect us against COVID-19. And some unique features about this vaccine is that it is made from mRNA, okay? And you may be wondering, what is a mRNA vaccine? What is it? Why is it important? And how is it different from traditional vaccines? And these mRNA vaccines, as of now, we have 2 that have been FDA approved for usage in our country to help treat this or to help protect us against this virus. And that is from Pfizer and Moderna. So they both essentially use the same technology. And so first, I'm going to go through 3 important concepts that are essential for you to understand and helping you to just get a better understanding on how these vaccines work. And so the first is just to understand that we have several main building blocks of life, okay? So in organisms, in all living organisms, there are these main macromolecules, is what we call them, that form the foundation of life. So first is carbohydrates, which are essentially your sugars. You have lipids, which consist of your fats, and proteins. And then lastly, which is of major importance for this particular vaccine, are our nucleic acids. Okay. And so your nucleic acids consist of your DNA and your RNA. So nucleic acids, particularly your DNA, are, and I like to say this to my students, is that your proteins are the physical manifestations of your genes. Okay. And so with that, your DNA ultimately goes into providing the code that informs our proteins that make up our body. So again, proteins are the physical manifestations of your genes. And to show that relationship, concept 2 is going to give you a better understanding of why that relationship is so important. And that is the central dogma of molecular biology, okay? And so what that central dogma is, and if you can remember back to your foundational biology years or studies, this should trigger some sort of memory, okay? And so what the central dogma says is that DNA is transcribed into messenger RNA, and messenger RNA is translated into protein. So here you see the relationship between DNA, messenger RNA, and protein. Because again, DNA, right, is going to make proteins, again, because protein are the physical manifestations of your genes, right? So you have DNA being made into RNA, being made into protein. And so RNA is sort of this intermediate step, right? And so it has the message of the DNA in a specific code that the protein process could read, okay? So this is important. This is essentially what messenger RNA is. It's the intermediate code or step in between the production of DNA and protein. So then the last important concept to know in order for you to get an understanding as to how these vaccines work is the basic composition of the membranes of cells. And so here you have a picture of a basic animal cell, okay? And if you can see, this red thing that wraps around it makes up what is known as the plasma membrane. And so here it is blown up. So you can see there's a lot of different proteins embedded into it, which is not of value to us right now. What's majorly of value is just to understand that this membrane has the ability to allow certain items in and out of it. And in the science world, we call that as being selectively permeable, okay? And one of the items that can easily pass through this selectively permeable membrane are fats, okay? So that's an important thing to note. Fats can readily pass through the plasma membrane of animal cells. Again, this is the third concept that is essential for you to understand in regards to how these vaccines work. So then let's put those 3 concepts together, right? So we know nucleic acids are one of the key building blocks to our to life. Then we know that the central dogma of molecular biology says that DNA is transcribed into RNA. RNA is translated into protein. And then lastly, we know that the composition of the plasma membrane of cells, or of animal cells specifically, is in such that fats can readily pass through. So those three concepts really form the foundational understanding as to how these mRNA vaccines work, okay? And I'm going to walk you through this process, and again, in a simplistic way without all of the heavy science jargon, okay? So here again, we have a picture of our coronavirus, okay? You can see the spike proteins that are found on the surface of its membrane, and then internally you see its nucleic acid. And again, because this particular strain of viruses use RNA as their nucleic acid, okay? And so what scientists did is they took only the messenger RNA sequence of the spike protein. They took that messenger RNA sequence and they inserted it into a lipid, okay? They're referring to it as a lipid nanoparticle just based on the size. And so now, based on those properties or those concepts that I've told you, this lipid nanoparticle is a fat, so it's able to readily pass through our cells, okay? And once it passes through our cells, this messenger RNA strand, okay, is now in our cell. This messenger RNA cell then results in our cell's ability to make the spike protein. That spike protein goes onto the surface of our cells, okay, or of this particular cell that has been impacted. That then triggers our body's immune system to identify this particular cell now in our body as being foreign. That results in the activation of our immune system and then ultimately the production of antibodies, okay? And it's these antibodies which is going to protect us against the virus if we ever come in contact with it again. And so just to back up again, so here is the vaccine. Again, the vaccine is just messenger RNA wrapped into a lipid. That lipid is able to readily pass into our cells, depositing now this messenger RNA sequence. And again, if you go back to the central dogma, the messenger RNA is now going to result in the production of a protein. In this case, that protein is going to be the spike protein of the coronavirus. That spike protein is now put on the surface of our cells. And because our cells don't naturally produce spike proteins, that's going to trigger the activation of specialized cells in our body to attack this cell and then ultimately result in the production of antibodies. And so it's important that I highlight some of the women that have really just been superheroes in regards to the production of this antibody and help create the technology. And so first, I want to shout out Dr. Caitlin Carrico. And she actually laid the foundation for the ability for us to develop these messenger RNA vaccines. And so she was actually a professor at the University of Pennsylvania on the tenure track. And she had a very difficult time securing grants to fund this concept. And so she actually became demoted. And because of that, just because of her struggles with just getting funding to support her ideas, she actually left academia and started working in the pharmaceutical companies. She refined the idea, and she now is currently the vice president of BioNTech, which is the company that worked with Pfizer to develop this vaccine that is essentially helping to subside this global pandemic. Then I also want to point out that Dr. Kismika Corbett, so she is a researcher at the NIH, particularly the Vaccine Research Center at the NIH, and she has done a lot of the foundational work with characterizing the genes and building the foundation for the messenger RNA sequence that was used in conjunction with Moderna to develop this vaccine. And so these are two women that are on the front lines that have really developed the foundational technology that is essentially helping to alleviate this situation. So I hope this short video was informative. And if you have any further questions, feel free to reach me in the department. And again, I'm Dr. Stacey Dutton. Thank you.